Okay, cool. Thank you for the honor of going last, Bob and Beth and Tony.
Anytime.
Yes.
We thought hard and long, and we said,
Yeah, no, I think it's because I show certain pictures that will elicit an eager thoughts of leaving. But in any event, I'm a psychiatrist, as many of you know, but I'm also a neuroscientist, but more importantly, I'm a wine enthusiast. And so rather than describe most of the talks I give solely in terms of psychiatric illness, I usually try to help people understand why wine is actually the key to my interests in neuroscience. So most emotions happen in anticipation of good things and bad things. And here's an example of a good thing. This is a rare bottle of Burgundy from a producer called Armand Rousseau. It's from the vineyard Le Chambertin, one of the great vineyards in Burgundy. If you're fortunate enough to have drunk this bottle with friends and family or been to the domain to taste it, just seeing this bottle, which in and of itself is not rewarding, can elicit all these pleasurable thoughts and feelings and anticipatory behaviors. So this bottle can make you feel really good if you're someone like me. On the other hand, if I'm invited to your house and I see this particular bottle on the table, I might get a feeling of dread. Why did I accept the invitation? So this is a different bottle with a different set of associations. It can make me feel quite bad and want to avoid the situation altogether. This was the motivating force behind the early work in my lab. And the early work in my lab was meant at looking at neural signals in the amygdala, trying to see whether neural signals in the amygdala were really specialized to anticipate bad things, because much of the literature in rodents had emphasized the role of the amygdala in aversive processing. There was some literature on appetitive processing. I think many people in the know believe that it played a role in appetitive processing, but in terms of neurophysiology of neurons and trying to demonstrate cleanly that there were specialized neurons for appetitive and aversive systems, that really hadn't been done. So we did an experiment. This was Joe Patton and Marina Belova and I did an experiment many, many years ago where monkeys would fixate, view a conditioned stimulus. These were novel fractals that had no inherent meaning. There'd be a temporal gap, a trace interval, and then we'd deliver an unconditioned stimulus. So this would be, for example, the first trial where the animal would view this stimulus and he would get a reward. Then he would get another stimulus. In this case, he'd get an aversive air puff directed at the monkey's face. Now, air puffs cause no tissue damage, so you can give it time and time again to the animal without actually causing any tissue damage whatsoever, but they don't like it. So it's unpleasant to them, and they will do things to protect themselves or avoid the stimulus at all. So it's an aversive stimulus, but one that one can safely use many times in monkeys. And in fact, people can use it in humans as well, and people have done human fear conditioning paradigms where the only aversive stimulus they use is a powerful air puff, say to the neck or to the face, and that's sufficient to condition cardiovascular responses and autonomic nervous system responses in humans, as well as in monkeys. So not that we're measuring, in this case, those autonomic measures. But in any event, there's an appetitive association and an aversive association, two different stimuli. The next trial might be another conditioned stimulus associated with aversiveness. Then there was a third one that was associated with nothing. And this would proceed until the animal had learned the associations. And we knew the animal had learned the associations because two behaviors emerged during this temporal interval, this trace interval, in anticipation of the US delivery. One was licking in anticipation of a liquid reward, and the other was blinking or eye closure in anticipation of the air puff that was directed at the monkey's face. All right, so at this point of the experiment, we could say something about the fact that the animal had learned those associations, but suppose we're recording from a neuron in the brain, and we get a differential response in response to this image compared to this image. You would have a problem on your hands at this point knowing what the meaning of that differential response was, because there's a confound here, is that the sensory stimuli are different for the two different CSs, and the reinforcement contingencies are different. And you can't tell then whether the neural responses that are different might be due to the different sensory stimuli or the different reinforcement contingencies. So what we would do is, without warning, on the fly, we all of a sudden would keep the stimuli the same but change the contingencies. Now the stimulus that was associated with something aversive was associated with something rewarding, and the other stimulus became associated with something aversive, and the animal would very quickly, within a few trials, learn these changed associations. And we recorded from single neurons in the monkey's brain, in the amygdala, while the animal was doing this task. And I'm just going to show you a couple of example neurons that really have formed the foundation for the last 10 or 15 years of work that's gone on in my lab. These are two different amygdala neurons recorded during one of these experiments. One is, we're showing firing rate on the y-axis and time on the x-axis. where the image comes on the screen at time 0, it goes off the screen at the second dashed line, and then we're showing you the trace interval right here. The US is delivered out here, so we're not showing the responses to the unconditioned stimulus here whatsoever. The left column is one neuron, the right column is another neuron. So when image 1 comes on the screen, the cell in image 1 is associated with reward. The cell gives you an elevated response during this trace interval, and image 3 initially is associated with something aversive, and the cell responds at a very low level. Without warning, the two flip, and within very few trials, the cell's responses flip. So now the cell is responding more to image 3 than to image 1, where it's responding low. So this cell responds more when an image is associated with something rewarding than when the same stimuli are associated with something aversive. We called it a positive cell because it preferred the appetitive association, and if that's all we had found in the amygdala, it would have been really quite surprising given the rich literature linking the amygdala to aversively mediated emotions. But it wasn't all we found. Roughly half the cells sensitive to reinforcement contingencies that we found in the amygdala preferred the opposite valence of reinforcement. And this is another cell. Here's image 1. When it was associated with something aversive, you give an elevated response compared to when the same image was associated with something appetitive. And here's the other image where it has the same valence preference. So this cell is preferring negative valence as opposed to this cell, which is preferring positive valence. We called these things positive and negative cells. They are anatomically intermingled in the amygdala, and so that you could sometimes even record from a positive cell and a negative cell from the same electrode tip in the same experiment. Subsequently, people have gone on to do experiments in rodents using activity-dependent expression of opsins to be able to access these cells that are anatomically intermingled and manipulate their activity. We're among them, and be able to show that, in fact, there's a causal relationship between appetitive networks and valence-specific behavior and aversive networks and valence-specific behavior in the amygdala. Now, one of the problems that we've become very concerned with in recent years is the idea that is the following, which is that, in point of fact, the relationship between stimuli and emotional significance in general, in our everyday lives, is not a one-to-one relationship. The same stimulus can have a very different meaning in different situations. And so therefore, you have to have ways in order to regulate your emotional responses to a stimulus depending upon the situation that you're in. And this is really the basis of the cognitive control of emotion. We apply it routinely without even thinking about it all the time. In different social situations, we can apply different rules for how we're going to respond to the same stimuli. We can feel differently about the same stimuli in very different situations. And the example that I often give about that in the context of my favorite sets of stimuli is this same bottle of Rousseau Chambertin. Now, when I was a student in Bill Newsom's lab many years ago, I actually could afford bottles like this, and it was really it was a wonderful thing. So if I nowadays encounter this bottle when I'm coming over to someone's house for dinner, and let's say it's someone's house that I've never been to before, so it's an entirely new situation. I don't have a representation of that situation, but I'm going over to dinner. I know I'm invited to a dinner party. I have the concept of dinner party in my head. I see this bottle on the dining room table. I instantly can assign it to be a positively valued stimulus because I have the concept of dinner party, and I instantly can categorize the set of stimuli there and say, boom, even though it's a new example of it, I know that I'm at a dinner party. It's a positive stimulus to me in that situation. I might see the same bottle in a wine store that I've never been to before. Completely new set of stimuli at the wine store. I know it's a wine store because I could recognize certain features that make me link it to the concept of a wine store, but I've never been to that wine store before. Now that same bottle, because it's so expensive, even though I'm a full professor, I can't afford it, I get this feeling of dread from the exact same stimulus because I'm encountering it in a wine store. And I can instantly assign that different value to the stimulus in these different situations because I have the concept of wine store and I have the concept of dinner party in my head, even when I encounter a new wine store or someone's house that I've never been to before. So this is reminiscent then of the problem that people talk about, which is often referred to as the curse of dimensionality in reinforcement learning. And the notion behind reinforcement learning is something like the following, is that there's a set of states in the world. These are all the different situations that you might be in. And then there's stimuli that you might encounter within those states, and you assign values to those stimuli. And the problem is that the states are unknown and can be huge. They're infinite, essentially, or virtually infinite. And so if you have to devote neural hardware to represent every possible state that you can encounter, you wind up with this very high dimensional problem. There's just not enough hardware there in order to represent all the different states. Instead, what you have to do is abstract the essence of certain features that link together different states according to a concept, like the concept of a dinner party or the concept of a wine store. This is what some people earlier in the meeting were referring to as an example of dimensionality reduction, where you would essentially reduce the number of states to some categorical representation of the types of states that they are, and then when you encounter a particular stimulus within one of those, you can instantly assign a value to it. But we really have no idea about how this process of abstraction or dimensionality reduction goes on in the brain. All the work that's usually done on reinforcement learning has to do with how do value-related signals, somewhat similar to the ones I already showed you, how do they actually operate? But we don't know much about how the brain can create representations of the states themselves, which really have to do with representing something like often concepts like dinner parties or wine stores, and often new concepts that you actually don't have in your head before. So this is a problem that we began trying to investigate in different ways really a number of years ago. This was our first take on that task and on that problem, and it's a very simpler task compared to the one that we're currently using, and there's some others that I'm not going to tell you about that are continuing this line of investigation. But in this task, we create two situations. We call them context 1 and context 2, and they're simply defined by different sets of reinforcement contingencies. And it's very similar to the task I already told you about. Stimulus A is followed by reward, stimulus B is followed by no reward in context 1, and the reinforcement contingencies flip in context 2, just like they were in the initial task. And these contingencies switch back and forth many times within an experiment. And in point of fact, the animals can only know which situation they're in by knowing the overall task set that they're in. So they'll know that in context 1, A is rewarded and B is not rewarded, and A is not rewarded and B is rewarded in context 2. And when they switch from one context to the other, they can experience one CS as having switched its reinforcement contingencies and then infer very quickly that the second one has also changed its contingencies. This suggested to us that the brain must have representations of these contexts, even though there's no explicit cue telling them which context they're in. There's just the sets of reinforcement contingencies. And our initial study on this looked at 3 brain areas that we were interested in relation to this kind of thing: the orbital frontal cortex, the anterior cingulate cortex, and the amygdala, where the original study that I showed you just a few minutes ago came from. We chose these in part because we knew we had these signals that were related to reinforcement expectation in the amygdala, and the OFC and the ACC were 2 brain areas from the prefrontal cortex that projected strongly down to the amygdala. And these are just 3 example neurons in the amygdala that in some ways presented the complexity of the problem before us, but also showed us that the signals there would be interesting to us. So what we're showing here is there's 4 different trial types: 2 from context 1, they're in solid lines, and 2 from context 2, they're in dotted lines. And we're showing you firing rate as a function of time again, where the image comes on the screen at the first vertical line and off the screen here, and this is the time of the US out here. In this OFC neuron right here, you can see there's differential activity during the time period before an image has even come on the screen. In this case, it's responding more in context 2 than context 1. This is not a stimulus-driven response. It's as if this cell has in its memory, I'm in context 2, not context 1. Then the image comes on the screen, and you can see the response properties of the cells are really quite complicated. During this initial burst of activity, it's actually responding to 3 out of the 4 stimulus trial types. And then if you go out here shortly before reinforcement, now it's actually giving you a signal that's telling you about which reinforcement you're going to get, because it's a solid blue line and a dashed red line, which means that you're likely to be, that you're going to be rewarded on the next trial. So this is a reward selective signal here. Here it's context selective, and here it's actually what Richard Anderson would have referred to as mixed selectivity, because it's responding to 3 out of 4 conditions. This is an ACC neuron that gives you context selectivity here and reinforcement selectivity here. Here's an amygdala neuron that's giving you context selectivity. Now it's responding to 3 conditions, and out here it's reinforcement expectation selectivity. So these neurons all exhibit what Richard Anderson was referring to as mixed and changing selectivity, and Stefano Fusi has done an extraordinary amount of work in terms of trying to understand how these neurons can contribute to different computational processes. And in fact, Stefano was a collaborator on this study, and he's a collaborator on the entire study, and really key collaborator on the whole rest of the work that I'm going to tell you about now. Now, this type of signal that I'm showing you here suggests that these cells and the networks of cells that are here could be representing something about the overall state upon which these values are assigned, but it doesn't actually tell us something about the nature of the representation. There are too few conditions within this experiment to really look at the nature of the representation in a really interesting way, because what we're interested in is this representation in a way that represented in these brain areas in a way that would support something that we call generalization. Richard Anderson referred to this very quickly the other day, but generalization is this property where if you have conceptual knowledge, which you build up by encountering many different examples of a certain type of thing, for example, many people's houses for a dinner party, and then you encounter a new house that you've never seen before, if you have stored in your brain information in a format having to do with dinner parties in the right format, even though you've never seen that new dinner party before, as soon as you see it, you can categorize it correctly. And we would say that your representation of dinner party in that situation is in an abstract format. It's because it's representing the conceptual information that you need in order to generalize to something new that you haven't seen before. And so we wanted to be able to design an experiment where we could start asking questions about whether representations of these abstract situations were present, whether they were present in an abstract format. And so what we have developed initially was a more complicated version of the task I just showed you before, and it's not more complicated in two ways. One way is that it now has an operant response. The monkey starts off by holding a bar, and then it sees an image, and now there's a correct operant response for this image. It's to release the bar in order to get a reward. So it's now an operant contingency and a reinforcement contingency here. And the other way this task is more complicated is that there's now 4 stimuli within a context, not just one. All of them start with holding the bar. 2 of them, the correct response is to release the bar. 2 of them, the correct response is to hold the bar. 2 of them are rewarded. 2 of them are not rewarded. Note that the operant contingencies are essentially orthogonal to the reinforcement contingencies because the correct choice, releasing the bar, doesn't tell you that you're going to get a reward. If you get one of the trials wrong where you're not going to get a reward, it repeats, so you can't just escape to a rewarded trial. So the animal is still incentivized to do the task, but he's got to do it correctly in order to proceed. Okay, so this is, now he's living in context 1, and context 1 now is not just defined by stimulus reinforcement contingencies, but it's defined by stimulus action or stimulus response outcome contingencies. So it's a whole set of contingencies here. The animal lives in this world, and then all of a sudden we change the world on him, and it will change some combination of the reinforcement and the operant contingencies. This is context 2, this is context 1. And then he'll switch back and forth between context 1 and context 2 many, many times. Now, I referred to the other task, the behavior in the other task. I referred to the idea that the animal uses inference. They also use inference on this task. I'll show you the data for that now. So in this task, if you look at the last stimulus before the animal experiences a context change, they're performing at a very high level, 95% correct, roughly. On the very first trial after a context change, they're performing at a low level. This is the first one here. This is uncued, so that's why they're performing so poorly. Now, if they were learning each stimulus type individually, that you would expect the next time they see another stimulus type, they'd still be performing very poorly. On the other hand, if they knew something about the structure of the task, then the next time they see a stimulus type, you would see an improved performance. And in fact, for the first stimulus type that they see on the first stimulus type that they see that's different, they're already performing above chance. And the next stimulus type, which is the third one, they're even more, and the fourth one, they're even more. They're not quite asymptotic all the way here, but so there's still possibly some learning going on, but this shows continued evidence of inference. And in fact, if you were to realign all of this data on the first correct inference trial that the animal shows, they're actually asymptotic from that point forward after every one of these switches. So they're clearly using something about the structure of the task to do this task. They're not just learning every stimulus response outcome individually. They clearly understand that there must be some kind of relationship between stimuli within these tasks, but how could that possibly be working? So now when they see stimulus A on a given trial, what could they possibly be doing? So here's stimulus A on the current trial. Should you release or should you hold? Now, if you're in context 2, stimulus A on the current trial may have been preceded by this sequence of trials, versus in stimulus A on this trial, it might have been preceded by this sequence of trials. And so one possibility is that all the animal does is memorizes the sequences of trials that he could have experienced and could use that kind of lookup table in order to perform the task. So if you had a B hold trial and then you see an A, you should know that you release. If you had an A release trial before and then you see an A, you should know you have a release. And here it's a different sequence of trials. And so if you have 4 possible stimuli here and 8 possible stimuli here, it's a lookup table with 32 entries and you could actually know what to do. That's not necessarily the most computationally efficient strategy, but looking at the behavior, it could account for the inference behavior that we just saw. The other possibility, though, is that the animal actually forms an abstract entity in its head, something like
a representation of the context that's defined by these sets of stimulus response outcome mappings. This is context 1, and this is context 2. And so if you just kind of collapse this all into a knowledge that these 4 trial types make up context 1, these 4 make up context 2. That's only 2 things you have to remember. 2 by 4 is 8. It's more computationally efficient, but now you're using a somewhat different strategy because you essentially have created a new hidden cognitive variable, this representation of context that's defined by these things. It's like this is the dinner party and this is the wine store, okay? How do you figure out which one of those strategies the animal's doing? Well, we can't actually look at the behavior and tell from the 2 because both of them would support the inference behavior that we can see. But we have an idea that we think we can look at the neural representations and ask which type of algorithm is the neural representation consistent with? Is there a representation in the brain that would support this kind of more higher level strategy where the animal has formed a new abstract hidden cognitive variable? And to do that, we've gone into the neural recordings. We've actually recorded from many brain areas on this different task. I'm going to tell you about recordings that we did in the hippocampus and 2 parts of the prefrontal cortex, the anterior cingulate cortex and the dorsolateral prefrontal cortex. Because when we initially saw those context-related signals in the brain that I showed you before in amygdala and OFC and ACC, we sort of started walking our way back through the brain to ask what earlier parts of processing might be generating these contextual representations. And that's why we went all the way back to the hippocampus and then 2 stages in the prefrontal cortex that we thought would potentially be important. We also, but not fully analyzed, have data from amygdala and OFC on this task as well. So what we're focusing in on here is a time period, the time period that I focused on before when I was showing you the context-related signals. It's a time period right before this stimulus B comes on the screen. And the reason we're doing this is the current trial B, is we want to ask what information is stored here. One possibility is that the information you're storing here is just related to a memory trace of the last trial. This was, say, an A release rewarded trial. And if that's all you're doing is remembering the last trial, that's good enough for a lookup table, but it's not good enough for this idea where you have abstracted the concept of context 1 or context 2. So how can we look at which one of those 2 things is this? Is this just a memory lookup table of the prior trial, or is it some kind of more abstract representation, a representation in abstract format? In order to look at that, I want to take you into something that I will call a firing rate space of neurons. And I'm going to show you this initially in 3 dimensions. This is imagining we're just recording from 3 neurons, but imagine if you have hundreds of neurons recorded from, you can do this in an n-dimensional space. And so what these 3 neurons are just this plot, F1 and F2 and F3. It's the activity of 3 neurons plotted against each other for stimulus A. This is somewhat similar to some of the plots that you saw earlier today. So this is what defined by the trial that came right before it. The yellow circle means it was rewarded. It's stimulus A when it's rewarded, and it gives you this pattern of activity between the 3 neurons, and we've just plotted it at a random location in this plot. In point of fact, for context 1, there are 4 different plots, 4 different data points, A, B, C, and D, all plotted at random points right here. And for context 2, there's 4 other points, and here they're all also plotted at random points as well. And in fact, if you were to look at all these points randomly located with each other, you can see that these 2 different contexts, the patterns of activity, are kind of entangled with each other. Now, it turns out, if you are recording from enough neurons, even though these things are somewhat entangled with each other, if you use these kind of linear decoders that Richard Anderson was talking about, and I think Andrew mentioned as well, it turns out if you're in a higher enough dimensional space, that is, you recorded from enough neurons, you can actually find hyperplanes that will allow you to classify which context you're in. So the way these decoders, generally speaking, work is that if you are plotting here, it's only 2 neurons, the activity of neuron 1 against the activity of neuron 2 for 2 conditions, say a plus condition and a minus condition. If you're just considering the activity of neuron 1, you can't really separate those 2 conditions from each other. But if you weight the neurons and then find a threshold, now you can. Imagine you're doing that in an n-dimensional space. That's essentially what these linear decoders are doing, weighting the neurons and finding an optimal hyperplane to classify. So if you're in a high enough dimensional space, you're going to be able to decode that context. And so in point of fact, when we apply that kind of decoder to hippocampal data, 650 dimensional space, 650 neurons, 340 from dorsolateral prefrontal, 402 from ACC, you can see we can decode context really quite well, even before the stimulus comes on the screen here. But this does not tell us that it's in an abstract format, because those data points I just showed you were all at random locations. And that's clearly just something that could be going about if it was just a memory trace of the last trial, with each last trial eliciting a different pattern of activity in the neurons. What would be an example of an abstraction where you're just remembering something, say, about the context? You would also be able to decode context just like that, but what might it look like? Well, the simplest example of that is what I would refer to as clustering. These are 4 data points from one context and 4 data points from another context. This is just simulated data. They're not all lying on top of each other because we've added a little bit of noise to this, but essentially these are well-separated points. They're not entangled like the other plot. You can see that we could decode context really well, but there is some loss of information about other variables because the neurons essentially are firing about the same way in the same patterns of activity for the 2 different contexts. So what's the problem? Why wouldn't the brain always represent variables in an abstract format by throwing everything out except for the variable that you care about and representing it in an abstract format by just creating 2 clusters? There is a fundamental problem with that I'm going to suggest that we have to be able to solve, and that fundamental problem can be illustrated simply by considering the fact that when you have this kind of clustering and you're throwing out all the information about the prior trial, say in this case, you're throwing out all the information other than it was a context 1 trial or a context 2 trial, you only can actually represent one abstract variable, and this will result in a great reduction in the flexibility you have in order to categorize things across different possible abstract variables. So the idea is that multiple concepts can actually be linked to single examples. So this is back to my favorite wine bottle again. Unfortunately for me, this can be linked to 2 different concepts, and as well as this one, which who cares? But in any event, this particular bottle is quite drinkable. That's a concept, but it's also quite valuable. So now it's unaffordable. That's 2 concepts linked to a single object. If all I do is represent something about really good versus really bad in these, when I'm representing this object, these 2 objects, I've lost information about value. This object here, well, that's not drinkable and not valuable. So I've linked 2 concepts to each of those 2 objects. How can you actually think of representing information in the brain in a way that you've represented things in an abstract format so that you can generalize the way we just were able to show you that clustering lets you generalize across context, but generalize across more than one variable? What I'm going to show you, our initial hint that this is what the brain may be doing in this task came from when we looked at data from the 3 brain areas that I'm telling you about, but we looked at it using a dimensionality reduction technique called multidimensional scaling. Multidimensional scaling you can think of as principal components, so essentially it's trying to preserve the distances between points as much as possible, but bringing everything down to 3 dimensions so that we can visualize the data as best we can. And when you do that, we're still capturing about 80% of the variance. So this is data from 640 hippocampus neurons reduced using this MDS technique down to 3 dimensions here. And this is the data from one context, this is the data from another context. The plus and minuses are telling you whether it's rewarded. The yellow also still means rewarded. And the H's and the R are holding and releasing the bar. So I'm going to rotate this data for you, and I think you can begin to see some structure to this geometry that indicates that there is still separation between the contexts, but there's not pure clustering, right? There looks like there's some organization within each context, but it's not that all the data points are lying on each other's, right on top of each other. This is the data from the hippocampus. I'm going to wheel through the 3 data sets here, and then I'll talk about it in a minute. Here's the data from the dorsolateral prefrontal cortex. Here, you can still see maybe even more within context structure, because it's starting to look like it's a plane within each of these 2 different contexts, but there's also separation between the 2. So if you look at the distance between the points within the context, they're really about as big as the distances between the contexts. That wasn't actually true in the hippocampus, but nonetheless, there's some structure here. It's not just random points. And when we go look at the anterior cingulate, it's even more pronounced. Here, things are looking like they're even more planar in form. And I'm going to stop the movie here at a point where I think I can emphasize, if I can get this thing back up. I think you can begin to see that there's how the rewarded trials are all lined up here, almost in one plane, non-rewarded here in another plane, and then there's 2 other different planes for the context here. And so this, looking at these plots rotate is what suggested to us that there's a way of reducing the dimensionality of these representations in the firing rate space in a way that's different than clustering, but in a way that will support the kind of generalization that I said was a signature of representing something in an abstract format. So here, when you move things within a context to all within a plane, that is dimensionality reduction. It's just not all the way down to a single point. And one way to do that is just to illustrate this schematically. These are the same 8 points plotted in each of these 2 plots. right? So this is context. This is where the blue means one context, the red means another context. Here are the yellow data points. That's rewarded. These are the non-rewarded ones. So this is 2 planes describing context, the same 8 points, 2 other planes, rewarded plane, non-rewarded plane. And so the notion here is that there's something in the geometry of the representations that allows you to represent multiple pieces of information in an abstract format. This will allow you to generalize across multiple variables and represent more than one variable in an abstract format. And we used 2 quantitative methods for being able to demonstrate that, in fact, this is what we are seeing in our actual data that I just showed you. Now, when we do these actual statistical tests that I'm going to tell you about, we don't do it in the MDS, dimensionality reduced space. We're doing it in the high dimensional space. So the statistical tests are all done in a high dimensional space. That was really just a visualization technique. And the 2 techniques we're going to talk about, one we call cross-condition generalization. This is actually the same technique that Richard talked about the other day, earlier today, because he was training on some trial types and testing a linear decoder on other trial types. And that's essentially an example of generalization. So imagine that we only have 2 points from one context and 2 points from another context. So this has simplified it down to 4. If you take a decoder and train it to weight neurons and find the best hyperplane for classifying context only looking at these 2 trial types, you can take that trained decoder and ask how well can it classify these other 2 trial types that it's never seen before. So that's analogous to my going to someone's house that I've never been to before and asking how well can I classify that as a dinner party, right? Because it's a new stimulus. This decoder hasn't seen it before. So if you are in a good you're representing things in a good abstract format, you should be able to train on some examples from a particular variable, in this case context, and test on another one and still be able to successfully decode it. We call this cross-condition generalization performance. Now, the plots I showed you before suggested that it was a particular geometry that afforded the ability to be able to do something like cross-condition generalization. And what we generated was a quantitative measure of that, which we call the parallelism score. And it turns out the parallelism score is very closely related to this cross-condition generalization, very closely correlated with this cross-condition generalization performance. And in the parallelism score, what we do is we look at a decoder, in this case trying to train a decoder to distinguish between A and C, determine its best hyperplane for decoding, and then calculate the vector that is orthogonal to that coding direction. That vector we call the decoding direction, and then we do the same thing for looking at trying to decode A versus C, training a decoder over here, calculate this vector and ask whether the coding vectors, these 2 different coding vectors, are parallel. And we do this for all possible combinations of points from two different values. So for example, context 1 and context 2, there's more than 4 points there. So we do it across all the different ones. So we generate essentially a parallelism score, which is asking how parallel are these coding vectors. And when you're in a high dimensional space, in order to get parallelism that's greater than what you would expect from some random model where you just shuffle the data points, is non-trivial. And we do the same kind of random model correction for the cross-condition generalization performance. So we can look at these representations in their high-dimensional format and actually ask what is the cross-condition generalization performance and what is the parallelism score. And we can ask it for each of 3 different task-relevant parameters that we looked at. One was context, which is the most important one in our case because that's sort of the hidden cognitive variable that we're asking whether the animal could be using in order to perform the task. But there are at least 2 other variables that we happen to know are stored during this time interval right before a conditioned stimulus comes on the screen. One is whether or not the animal received a reward on the last trial, so that's the reward of the last trial. And the other is whether the animal happened to release or hold the bar on the last trial, that's the recently performed action. And those 3 things together tell you about what the last trial was. So we're going to ask whether information about the context, as well as information about the reward value, as well as information about the action, both of the last trial, are represented in an abstract format. When you look at the parallelism score, what you can see is that in the hippocampus, the context parallelism score is very significantly greater than the random model predicts. This is a 95% confidence interval on a random model of this, where you shuffle all the data points. The value is also stored in a very abstract format, but the recently performed action is not. When you look at the dorsolateral prefrontal cortex, all 3 variables are stored in an abstract format, and the same is true in the anterior cingulate cortex. And I think you could see some of this start to emerge if you would look closely when I was showing you those movies, because you got more and more of a planar format, and the points having to do with these different variables were better and better aligned with each other in these planar formats. So this is the parallelism score all variables in abstract format in prefrontal cortex, 2 out of 3 in the hippocampus. For cross-condition generalization, you have a choice to make. How many of the conditions do you test on versus how many do you try? How many do you train? I mean, how many do you train on versus how many do you test? Here we're training on 3 out of the 4 conditions for each dichotomy and testing on 1. The results are similar if we train on 2 and test on 2, etc. So we train on those 3 and test on those 2. And when you go do that in the hippocampus, you get results that match the parallelism score, the same in dorsolateral prefrontal cortex and the same in anterior cingulate cortex. These are all significantly above chance. The action in the hippocampus is not. Now, having the representation be in an abstract format supports generalization, as indicated by this cross-condition generalization performance. And when you fail to see the information in an abstract format, it does not mean that the information's gone. It just means it hasn't been put in a format that's sort of dimensionality reduced so that you can do this kind of cross-condition generalization. So if you just try to do straight decoding analysis of the last trial, this is going to be training on a subset of all the trials and testing on held-out trials. You can actually decode at above chance levels. And it's a weaker signal for action in the hippocampus, but there's still information about the recently performed action in the hippocampus. So all these variables look like they're, to some degree, stored in an abstract format, except for the action in the hippocampus. All right, let me try to wrap up. I know I raced through a lot. So neurons, this is an old study, neurons in the amygdala represent expected, rewarding, or aversive stimuli. And these neurons, we know from work in my lab and others, mediate expression of valence-specific emotional behavior. However, the cognitive regulation of emotion often refers to the ability to gate emotional responses according to one's situation or context, even when context is uncued. And uncued contextual information is represented in an abstract format, a format that supports generalization in hippocampus, ACC, and dorsolateral prefrontal cortex. Preliminarily, I think it's also there in an abstract format in amygdala and OFC, but we haven't finished analyzing it to be sure. In point of fact, it's actually multiple variables that are represented simultaneously in an abstract format in all the areas, with all 3 variables stored in an abstract format in the prefrontal cortex, and 2 out of 3 in the hippocampus. And this is accomplished essentially by reducing the dimensionality of the representation so that you can still preserve information about the task-relevant parameters, but put it in a format in the firing rate space that will enable you to generalize to new situations when you encounter them. And this whole one of our big ideas behind this is that people often have investigated brain areas trying to ask what information is represented in the brain. Do you see single cells that represent the information? Can you look at the population and decode the information? This is asking a different question. This is asking a question of whether the information is represented in a particular format that will allow generalization, so that when you encounter a new example that you haven't seen before, can you generalize to it? And that is exactly what's needed to demonstrate application of the fact that you have conceptual knowledge. And in point of fact, we all need to be able to form new concepts, form new representations that represent those concepts, and to be able to generalize those concepts to new situations. That's what we need to do in order to be able to regulate our emotions. And in point of fact, we have a very limited information about how the brain actually can construct representations of new concepts that would support this kind of generalization. And I would suggest that these kinds of methods may well be key for asking a new level of questions about how brain areas differ. Not just asking whether the information is represented, but asking something about in what format is the representation there, and therefore could it support the kind of generalization that is going to underlie the cognitive regulation of emotion. All right, so I didn't do any of this work. The initial work on context was done by Alex Saez, Jerome Minora, and Sylvia Bernardi did all the work on the last big part of the task that I told you about. Brian Lau and Chris Peck were key members of the lab that participated in lots of different aspects, and Marina and Joe did all the original experiments, and I'm forever grateful to them. Conceptually and analytically and in every way, I'm enormously indebted to Stefano Fusi and the people in his lab, Mattia Rigatti, who's now at IBM, and Marcus Benna. I didn't tell you about, there's a whole modeling section of the story. So when we train shallow deep networks, which say just 2 hidden layers on solving new problems, we observe the same geometry that we've just described in the neural representations. We see that same geometry in those networks as well. So we think this may be a very fundamental principle about how the brain forms new conceptual information that enables generalization. Thank you, and we can go enjoy some red burgundy.